Good morning, everyone, and thank you for joining the H.C. Wainwright BIOCONNECT 2022 conference. My name is Chet, and I'm a Biotech Equity Research Associate here at H.C. Wainwright. While we are virtual this year, we're confident that we're going to be able to provide value to you with over 550 companies presenting at this conference, as well as via your interactions through one-on-one meetings. H.C. Wainwright is a full-service investment bank dedicated to providing corporate finance, strategic advisory, and related services to public and private companies across multiple sectors and regions. We have a total of 23 publishing senior analysts and over 630 companies covered across all sectors. Please visit hcwco.com for more information. From a logistics standpoint, please make sure to reference your BioConnect Virtual Conference online portal that provides your individual links to your meetings and all presentations. Please join us for corporate presentations and panels that will be available live and streamed from January 10th to the 13th. With that said, have a productive and enjoyable day, and I'd like to introduce our presenter. I'd like to welcome Jonathan Drachman, who's the CEO of Neoleukin Therapeutics, a biopharmaceutical company focused on de novo protein development, leveraging computational methods. Over to you, Jonathan.
Chet, thank you. Thanks for the introduction, and thanks for the invitation to present at the H.C. Wainwright BioConnect conference. It's a pleasure to be here. I look forward in the next 20 minutes to telling you about Neoleukin Therapeutics. This is our forward-looking statement for updated risk factors and other disclosures that are made periodically to the SEC. Please see our website. Neoleukin Therapeutics was founded in 2018 based on computational technology that allows the company to design completely de novo proteins. And we are targeting making cytokine mimetics, both to activate or inhibit the immune system. The lead molecule, NL-201, is a highly potent non-alpha both IL-2 and IL-15 agonist that is being developed for cancer immunotherapy. In 2019, the company went public through a merger. In 2020, we submitted the IND for NL-201, and in 2021, we began the first-in-human clinical phase 1 testing. We look forward in 2022 to talking about how we are broadening our strategy, both with NL-201 and with additional molecules that we've been developing in research. In 2021 was a busy year for the company. We moved into new headquarters in Seattle, Washington, with a new lab and office space. We started our phase 1 clinical trial in solid tumors. We announced plans to begin a second trial this year in hematologic malignancies. We presented data on a second molecule, which is an inhibitor of the IL-2 and IL-15 pathways, potentially to be developed for inflammatory conditions. We broadened our executive leadership team, adding a chief medical officer and head of research. And we continued to build out the company in other areas, including CMC and the clinical department. And we're now approaching 100 employees. This is our leadership team with a lot of deep experience in drug development and in corporate operations. So I'm going to start by talking about the lead molecule, NL-201, and the properties that we believe make it a very exciting drug for the development in immuno-oncology. NL-201, as I mentioned before, is fully de novo. It has no binding to the alpha subunit of the IL-2 receptor. So it is actually a pure beta and gamma agonist. Because it has no residual binding to the alpha subunit, there's no bias towards cells that express the alpha subunit, such as T regulatory cells or endothelial cells. It's more potent than either native IL-2 or IL-15 on the immune cells we want to activate. And it activates both through both pathways, CD8 T cells and NK cells. It's also designed to be very thermodynamically stable and to be very compact and hydrophilic, to have good drug-like properties. The reason why we wanted to design a new de novo IL-2 molecule is that the native IL-2 molecule has some significant properties that make it a non-ideal pharmacologic agent. The most important of those is that the alpha subunit directs IL-2 preferentially to cells that express CD25, and that includes endothelial cells, T regulatory cells, and other immune cells. So at low doses, IL-2 ends up causing proliferation of these immunosuppressive cells, and at high doses, it can cause damage to blood vessels and other organs. So we wanted to create a molecule that would have more binding to beta and gamma. We did that through looking at the crystal structure of IL-2 bound to the alpha, beta, and gamma subunits, identifying the critical interfaces on the beta and gamma subunit, and then eliminating all potential binding to the alpha subunit, using our computational approaches and algorithms to design a completely new topology that would enable this beta gamma binding, but lack the interacting domain with the alpha subunit, and then look at substituting any possible amino acid at those positions to optimize the stability and interaction with the beta gamma receptors. The resulting molecule, Neo215, had less than 15% conserved sequence compared to native IL-2, but as you can see here, forms a very compact 4-helix structure and binds tightly to the beta and gamma subunits. When looked at in vitro with human cells, you can see in this slide that NL-201 is more potent at activating signaling on naive CD8 T cells and NK cells 3 to 10 times more potent than native human IL-2, but it's 30 times less potent at activating T regulatory cells. The result of that is when you look at a ratio of the proliferation on CD8 to T regulatory cells, you can see that at low concentrations, NL-201 is highly proliferative, causing significantly more proliferation in the T cells than in T regulatory cells, whereas IL-2 is actually immunosuppressive with a ratio less than 1 at low concentrations. And we see similar things with NK and Treg proliferation. In vivo, we've shown that NL-201 is very potent and well tolerated when given weekly ×2. This is a colon cancer model, CT26 in mice. We show that 2 doses causes minimum weight loss. It has a significant impact on delay in tumor growth and completely eliminating tumor in some animals. And then when animals where the tumor has gone away are re-challenged with the same tumor, they fail to implant. We looked at 12 different syngeneic tumor models across a wide spectrum of tumor types, ranging from GI malignancies to lymphoma, prostate, lung, breast, kidney, and melanoma. And in every case, we saw statistically significant inhibition of tumor growth from 2 doses of NL-201 compared to control. We performed our GLP toxicology study on non-human primates with 5 weekly doses. And one of the things that we wanted to look carefully at was the potential for a de novo protein to be immunogenic in non-human primates. Here we saw that after the 5 doses, only 4 of 26 animals developed detectable anti-drug antibodies, and those were all at the low or moderate positive control level. We also wanted to see what the pharmacodynamic effect was of both NL-201 as well as of those few animals that developed anti-drug antibodies. Those are depicted here in pink, and you can see that after the antibodies had formed, when the 5th dose was administered, those animals who had detectable anti-drug antibodies had no different pharmacodynamic pattern than those that were negative. I'll also point out that at all dose levels, including at the lowest dose level, we saw persistent proliferation of CD8 T cells for at least 7 days with a single dose. So in the middle of 2021, we began the first in human clinical trial with NL-201. This is delivering NL-201 intravenously, systemically, as monotherapy in patients with relapsed and refractory solid tumors. Part 1 of the study is designed to be a dose escalation, looking at 2 different schedules, either given once every 3 weeks or twice every 3 weeks, to identify the optimal dose and schedule and tolerability for future development. Once we have identified that recommended phase 2 dose and schedule, we will begin part 2, which includes indication-specific expansion cohorts, including in renal cell cancer and melanoma. This is being conducted at multiple sites in Australia, the United States, and Canada, and we expect to have enrollment of up to 120 patients, and we're planning to present interim data this year. The NL-201 has a very broad opportunity for use in cancer. We, in addition to the solid tumor trial that began last year, we plan to begin a trial in patients with hematologic malignancies this year based on data that we have developed internally, as well as data that was presented at the ASH conference in December of 2021, and historical data on the role of IL-2 and IL-15 in hematologic malignancies. We also have shown preclinically that NL-201 is quite active when combined with other standard of care agents, and we think there's great opportunity for novel therapies where NL-201 could be a critical backbone of new regimens. And there's the potential to administer NL-201 locally, as we showed at SITC last year. One of the recent presentations of preclinical data showed that when NL-201 is given systemically in a cold tumor, the B16 melanoma model, not only are we able to increase the number of T cells in the tumor microenvironment, but also the diversity of those T cells, and increase the number of different T cell clones that are present in the tumor. And that's something that distinguishes it from checkpoint inhibitors, for example. We also showed that alone and in combination with checkpoint inhibitors, we see good increases in interferon gamma, granzyme B, and decreased tumor growth in this in vivo model. We've also shown that NL-201 can potently increase PD-1 expression on T cells at very low concentrations. And that, of course, increases the possibility of NL-201 combined with checkpoint inhibitors. And we showed that with multiple models here, breast cancer model and kidney cancer model that are relatively refractory to checkpoint inhibitors, we're able to see good synergy when combining the checkpoint inhibitors with NL-201. Previously, we've shown that NL-201 also can have great additive or synergistic activity when combined with CAR T cells, shown here with good tumor reduction and also tremendous expansion of the CD8 CAR T cells in the tumor microenvironment, as well as when combined with antibodies, in this case, on the right, a TA99 antibody, which by itself has little activity, but when combined with NL-201 has greater tumor control than either separately. And then, as I mentioned last year, we also showed that local administration of NL-201 by direct injection into a tumor on one flank can have good effects on control of the tumor on the opposite flank. So if you inject the right flank, the left tumor on the left flank is also delayed in growth. And there's better tolerability at high doses, where we can see similar tumor growth inhibition compared to IV administration, but less weight loss and toxicity. One of the nice things about de novo protein design is that the proteins are extremely able to be changed and modified. We can change the half-life, we can change the affinity, we can fuse or conjugate them to targeting domains, and they can be made conditionally active. And those are all things that we're working on in research. We disclosed at the ACR conference last year a novel molecule, Neo5171, that is an inhibitor of innate inflammatory IL-2 and IL-15. It works by binding very tightly to the IL-2 receptor beta, but having no potential to bind to the gamma chain. So it disrupts the signaling of beta and gamma. And you can see here at low subnanomolar concentrations, it can inhibit the proliferation that's induced by 1 nanomolar of IL-2. When given systemically with extended half-life, we show that this has a potent effect on improving survival of animals in an aggressive graft versus host disease model. And as far as conditional activation, we've previously shown that one advantage of de novo proteins could be the ability to divide the molecule into two pieces, neither of which can interact with the receptor, but when brought together, can restore signaling. And then if both of those pieces, part A and part B, are targeted to the tumor, then we can restore delay in tumor growth and see some animals that are even tumor-free without seeing any toxicity or as evidenced by weight loss. So our founders are leaders in the de novo protein design emerging field. We have published several papers, including in 2020, on developing an inhibitor of infection by SARS-CoV-2. This was by creating a mimic to the ACE2 human receptor that would outcompete the virus and prevent infection. And there's a cartoon here depicting how this is done. We designed NLCVX1 to bind tightly to the spike protein, exactly mimicking ACE2 and preventing the virus from being able to enter human cells. And this was published in Science at the end of 2020. Finally, our financial highlights and upcoming milestones. We finished the third quarter of 2021 with just under $155 million in cash, which is expected to fund our anticipated operations into the second half of 2023. We currently have 42.4 million common shares outstanding and an additional 12.7 million pre-funded warrants. In 2022, we expect to present the first interim phase 1 data of NL-201 and initiate a new trial in heme malignancies with NL-201. And with that, I want to thank you all for your attention, and I look forward to meetings throughout the conference and answering your questions. Thank you very much.
Awesome. Thank you so much, Jonathan. In the interest of time, I'll wrap this up, but I do have one quick question. Looking at some of the slides that are focused on checkpoint inhibition and a potential combination, are you also looking at patients who are resistant or who develop resistance to checkpoint inhibitors as a potential target population? either in combination or maybe even as a follow-on therapy.
Yes, I think that could be a very interesting area because the checkpoint inhibitors are more like taking the brakes off the immune system, but what we are doing is actually activating the immune system. And as we showed, taking cold tumors, increasing the number and the variety of infiltrating T cells. And we think that that could be a really nice way of turning cold tumors hot and enabling them to respond to checkpoint inhibitors in the future.
Gotcha. All right, great. Thank you so much. I want to thank our presenters for taking part in what has been a very productive and informative series of presentations. We appreciate the time and effort that went into preparing them. Hopefully, our next conference will be one that we can hold in person rather than virtually. But in the meantime, we're very grateful for your flexibility and your presence online this year. Thank you again from the H.C. Wainwright team.